I spent 15 years running DNA analysis on samples that were not supposed to exist, and the day I finally understood what I was looking at, I burned my own career to the ground rather than hand the data to the people who wanted it. I was a wildlife geneticist at the University of Northern British Columbia. I had a lab, a tenure track position, and a reputation for being the most conservative, most boring, most by the book scientist in my department. That reputation is precisely why a retired trapper walked 200 kilometers out of his way in 1996 to bring me a sample instead of taking it to the newspapers. He knew I would not sensationalize it. He did not know that I would spend the next decade and a half of my life quietly confirming that everything he suspected was true, and that the confirmation would eventually cost me my job, my funding, and very nearly my marriage. Before we get into it, let me know where you're watching from and if you've had an encounter like this. My name is Dr. Wallace Kinney. I am 69 years old now, retired, living outside Terrace, British Columbia, in a house my wife Joan and I bought in 2012, the year everything ended for me at the university. I earned my doctorate in molecular genetics at the University of British Columbia in Vancouver in 1983, under a supervisor who specialized in population genetics of coastal salmon runs. I spent the following decade working for the Provincial Ministry of Environment, doing exactly the kind of unglamorous, methodical genetic fieldwork that nobody outside the field ever hears about. Grizzly bear hair snag studies, wolf scat analysis for pack territory mapping, salmon fry genotyping to track hatchery contamination in wild stocks. I was good at it because I did not get excited about anything. My colleagues used to joke that I could find a two-headed calf in a barn, and my first instinct would be to check the lab's chain of custody paperwork before I checked the calf. In 1994, the University of Northern British Columbia opened its doors in Prince George, the first standalone university built in the province's north in living memory. I was hired the following year to help build out their new molecular ecology program, and by 1996 I had my own lab, two technicians, and a caseload that came almost entirely from ministry contracts and graduate research. Prince George sits at the confluence of the Fraser and Nechako rivers, roughly the geographic center of the province, and from there, the university's research reach extended out into some of the most remote country in North America. The Skeena watershed to the west, the Omanika Mountains to the north, the Bulkley Valley with its logging towns and its ranch country and its long, cold winters running 200 kilometers west of Prince George along Highway 16 toward Smithers and Terrace, and eventually the coast. That first year, I ran the kind of samples every wildlife genetics lab in the country runs. Hair caught on barbed wire snag stations set up to census grizzly populations, scat collected along cut block edges to study wolf diet composition after a logging operation changed the local prey base, tissue punches from ear-tagged moose calves for a herd health study funded jointly by the ministry and a consortium of forestry companies who needed the data for their environmental assessments. It was slow, careful, unglamorous work, and I loved it precisely because it was unglamorous. There is a piece in work that nobody expects to be interesting. The sample that changed my life arrived in October of 1996, delivered to my office door by a man named Raymond Joseph. Raymond was a Wet'suwet'en trapper and guide outfitter in his early 60s who had run a trap line in the Telkwa Range, south of Smithers, for most of his adult life, a stretch of country that climbs from the Bulkley Valley floor up through subalpine fir and into rock and ice above the tree line. He had heard about my lab through a ministry biologist he had worked with on a Fisher reintroduction study 2 years earlier, and he had driven from Smithers to Prince George nearly 4 hours each way, specifically to hand me a resealable plastic bag containing a clump of dark hair he had pulled off a strand of barbed wire he used to mark the boundary of one of his trapline cabins. He did not tell me what he thought it was. He told me where he found it, the date, the weather conditions, and the approximate size and depth of a set of tracks he had found nearby in fresh snow 2 days earlier, tracks he had photographed with a disposable camera and measured with a carpenter's tape. 16 inches long, stride length of nearly 6 feet in a walking gait based on the spacing. He was matter-of-fact about it in the way that people who spend their entire lives outdoors tend to be matter-of-fact about things that would send other people running for the nearest exit. He said, I'm not asking you to tell me what it is. I'm asking you to tell me what it isn't. I told him I would run it through the standard panel we used for hair identification, mitochondrial DNA sequencing targeted at the cytochrome B gene, which is the workhorse marker for species identification because it varies enough between species to be diagnostic, but is conserved enough within a species that you get clean, reliable matches against reference databases. I told him it would take a few weeks given everything else in the queue. He thanked me, shook my hand, and drove back to Smithers the same afternoon. I ran the sample in November. The extraction was clean, the sequence quality was good, no signs of degradation or contamination that would compromise the read. I ran it through the standard comparison database we used at the time, which cross-referenced against known mammalian species common to British Columbia: black bear, grizzly bear, moose, elk, though elk were not native to that specific drainage, mountain goat, wolf, cougar, human for contamination screening, which is standard procedure because human DNA contamination is the single most common source of error in field-collected hair samples.
Quick thing before we get into it. I have a book coming out. It's called the Bigfoot Codex. It's a compilation of all my research and findings. It's kind of my baby. So if you want to check it out, links down below. And let's get you back to the video.
None of them matched. Not partially, not close enough to suggest degraded sequence or a poor quality read. The sequence simply did not correspond to any entry in the reference database I had access to. My first assumption, and I want to be honest with you about this because it is the assumption any competent scientist would make, was that the sample was contaminated or that I had made a technical error somewhere in the extraction or amplification process. I re-ran the extraction from a second hair in the same bag. Same result. I ran a blank control alongside it to rule out reagent contamination. Clean. I sent a third hair to a colleague at the University of Alberta who ran a separate lab with different equipment and different reagent lots, specifically so that any systematic contamination in my own lab would not carry over. He called me in January of 1997 and told me his sequence matched mine almost exactly and that he had no idea what he was looking at either. I called Raymond Joseph and told him the results were inconclusive. That was true in the narrowest technical sense. It was also the first time in my career that I chose to shade the truth rather than report it plainly, and it would not be the last. I want to explain something about how a genetics lab actually works, because it matters for understanding what happened over the following years. A cytochrome B sequence that does not match anything in a reference database is not, by itself, evidence of anything exotic. Reference databases are incomplete. There are thousands of documented mammal species and countless subspecies and regional populations whose full mitochondrial genomes have never been sequenced and deposited anywhere. The honest, responsible interpretation of an unmatched sequence is that you are looking at a species or possibly a subspecies that is undersampled in the literature. It happens more often than people outside the field would guess. My job, as I understood it in 1997, was not to speculate about what the sample might be. My job was to keep collecting data until the pattern told me something I could defend. So that is what I did. I told Raymond that if he found anything similar again, I would be interested in running it. I did not tell my department head about the anomalous result, because at that point it was one unmatched sequence out of hundreds of routine samples I processed that year, and flagging it would have meant writing a report that said, in essence, I found something I cannot explain, which is not the kind of thing a junior faculty member wants attached to his name in his second year at a new university trying to build a reputation for rigor. Raymond came back in April of 1998 with a second sample. This one was different in kind. It was not hair caught on wire. It was a fecal sample he had found near a game trail at roughly 4,000 feet elevation in the same general drainage, a location he described as being well above where he would expect to find bear sign that time of year, since the local grizzly and black bear populations were still largely denned up or just beginning to emerge at lower elevations. The scat itself was unusual in composition. It contained a mixture of plant material, including early spring shoots and inner bark scraping consistent with cambium feeding, along with what appeared to be small mammal bone fragments, suggesting an omnivorous diet with both herbivorous and carnivorous components in early spring before most fruiting plants would have been available. That dietary pattern alone was not unheard of. Black bears in early spring will eat almost anything, including carrion and small mammals, when vegetation is scarce. What was unusual was the diameter and volume of the sample, which by visual estimate exceeded what I would expect from either a black bear or even a large male grizzly at that time of year, when both species are typically thin and their digestive output correspondingly modest after a long winter's fast. I ran the DNA extraction from the fecal sample using the standard fecal DNA protocol, which is more technically demanding than hair extraction because fecal samples contain a much higher ratio of bacterial and plant DNA relative to the host animal's own genetic material, requiring more careful primer selection to isolate the target sequence. The mitochondrial sequence I recovered was a 94% match to the sequence from Raymond's original hair sample 2 years earlier. Not identical, which made sense if the samples came from two different individuals within the same population, but close enough to confirm they belonged to the same undocumented lineage. That was the point at which I stopped thinking of this as a curiosity and started thinking of it as a research question. I want to be precise about what that meant in practice, because it is easy for people outside science to imagine that a moment like that comes with some kind of dramatic realization. It did not. It came with a decision to allocate more of my limited research time and my limited grant funding to a line of inquiry that had, at that point, produced exactly 2 data points from a rural trapper with no formal scientific training. I told myself I was doing this because unmatched mitochondrial sequences from the same general region, replicated across 2 independent samples collected 2 years apart, constituted a legitimate, if minor, biodiversity question. An undocumented population of something. Possibly a hybrid wolf-dog lineage with unusual mitochondrial haplotypes, which does happen in areas where feral or free-ranging dogs interact with wolf populations. Possibly a genetically distinct, geographically isolated bear population that had diverged enough to produce an unusual mitochondrial signature, which is biologically plausible in mountainous terrain where populations can become isolated by topography. I did not in 1998 allow myself to consider the explanation that a certain segment of the public would have jumped to immediately. I was a tenure-track scientist at a new university trying to establish a molecular ecology program with real credibility in a province full of forestry companies and government ministries who needed to trust my data for regulatory and environmental assessment purposes. The word Sasquatch did not appear in my notes for another 4 years, and even then it appeared in quotation marks in a private notebook I kept separate from my official lab records because I understood even then that the word itself was radioactive to a scientific career, regardless of what evidence might eventually justify using it. Over the next several years, from 1998 through 2002, I built a quiet, unofficial side project around this question, running alongside my funded research rather than displacing it. Raymond continued to bring me samples when his trapline work took him through the upper Telqua drainage, usually once or twice a year. I began cautiously to ask him more about what he had observed over his decades in that country, and he told me things that a career field scientist learns to take seriously even when they arrive without physical evidence, because experienced backcountry people are, as a rule, extremely conservative in what they report. They do not want to be laughed at any more than I did. Raymond told me about vocalizations he had heard at night from his cabin, low, sustained calls that did not match any owl, wolf, or cougar, sound he had cataloged across 40 years in that country. He told me about finding trees with bark stripped in vertical strokes far above the reach of any bear standing on its hind legs, strokes that appeared to have been made with something narrow and hard, possibly a claw or a tool, at heights approaching 9 feet from the ground. He told me about food caches he maintained for his trapline operation that had been raided in ways inconsistent with bear behavior, with containers opened rather than torn, lids removed rather than clawed off. I want to be careful here, because this is exactly the kind of testimony that, presented uncritically, undermines scientific credibility rather than building it. Anecdotal reports of unusual sounds and unusual sign are not evidence in the way that a DNA sequence is evidence. I treated them the way any careful scientist treats anecdotal field observation, as a source of hypotheses to test, not as conclusions in themselves. What kept me engaged was not the stories, It was the fact that every time Raymond brought me a new sample, the DNA results kept confirming the same undocumented lineage, and the geographic pattern kept narrowing toward a specific stretch of high country in the Telkwa Range, roughly bounded by Hunter Basin to the north and the headwaters of Buck Creek to the south, an area of perhaps 60 square kilometers of subalpine and alpine terrain that saw almost no human traffic outside of Raymond's own trapline circuit and the occasional backcountry hiker in midsummer. By 2000, I had 6 independent samples, 4 hair and 2 fecal, all sequencing to the same mitochondrial lineage with a level of internal variation consistent with a small breeding population rather than a single individual. I brought in a second colleague at that point, a population geneticist named Dr. Priya Chandra at Simon Fraser University, who specialized in estimating minimum viable population sizes from limited genetic sampling. I sent her the sequence data without telling her where it came from or what species I suspected, framing it as an unidentified carnivore population of conservation interest, which was not, strictly speaking, a lie. She ran the numbers using standard population genetics software and came back with an estimate that the samples represented a minimum of 4 to 7 distinct individuals based on allelic variation patterns, with a caveat that 6 samples was a small data set and the true population could be considerably larger or less likely could represent a population bottleneck with fewer founding individuals than the sample diversity suggested. I did not tell Priya what I suspected in 2000, and for a long time I assumed that First Exchange would be the only contact we ever had on the subject. That assumption turned out to be wrong, and the way it turned out to be wrong is worth telling because it shaped how I thought about disclosure for the rest of the project. Priya reached out to me again in 2004, unprompted, having apparently kept the unusual dataset in the back of her mind for 4 years, the way a good population geneticist keeps any unresolved anomaly in the back of her mind. She asked in an email I still have printed and filed away whether I had ever managed to identify the species behind the samples she had analyzed and whether I would be willing to share any additional data, since she had in the intervening years become increasingly interested in a methodological question about how minimum viable population estimates degrade in accuracy when the founding sample size is small and the species itself is entirely unclassified. A scenario she said came up more often in conservation genetics than most people appreciated, particularly for newly discovered or newly rediscovered species where no prior population data existed at all. I did not tell her the full truth even then, though I came closer than I had with almost anyone outside my own small circle. I told her carefully that the samples were from an undocumented hominid lineage using the same dry clinical framing I had settled into with Colin the following year, and I watched, over a series of increasingly frank emails through the fall of 2004, her own skepticism give way slowly and reluctantly to a kind of guarded scientific curiosity that mirrored my own progression 8 years earlier almost exactly. She asked for the raw sequence data. I sent her an anonymized subset stripped of any collection location beyond a generic North-Central British Columbia designation, and she ran her own independent population modeling using a set of statistical assumptions different from the ones I had used with my original analysis, arriving at a minimum viable population estimate that was, if anything, slightly more conservative than mine, suggesting a total population across whatever range the samples represented of no fewer than 12 individuals and quite possibly several times that number, depending on how much genetic structure existed between subpopulations that my limited sampling had not yet detected. Priya became, over the following years, an occasional and extremely careful correspondent on the project, never told the specific locations, never brought fully into the operational side of the research the way Raymond, Colin, Joan, and eventually Trevor were, but consulted periodically on statistical questions that required expertise I did not have, and she was, in the end, one of the 2 anonymous peer reviewers whose comments strengthened my eventual 2012 manuscript, a fact I only learned years later when she mentioned, almost offhandedly over coffee at a conference in Victoria, in 2015 that she had recognized the dataset the moment the manuscript crossed her desk for review and had recused herself from formally disclosing that recognition to the journal on the grounds that her prior informal involvement did not rise to the level of a conflict of interest requiring disclosure, a judgment call I have never been entirely certain was correct, but that I have also never had any inclination to question, given how much her careful, rigorous engagement with the data over nearly a decade had strengthened the final published work. I did not tell my department head about the project at all during those first years, which in retrospect was a mistake, though I understand exactly why I made it. University research funding operates on cycles of grant applications, peer review, and institutional oversight, and any project framed around cryptid investigation, however carefully worded, would have been laughed out of every funding committee in the country and would likely have damaged my standing with the funded research I was doing at the time, which included several legitimate publishable studies on mountain caribou genetics that mattered a great deal to provincial wildlife management policy. I compartmentalized. I ran the side project on my own time, using leftover reagents from funded projects in lab time during evenings and weekends, and I told myself that if the pattern ever became strong enough to justify a formal research proposal, I would find a way to present it that would survive institutional scrutiny. I should describe, briefly, what the actual laboratory work looked like during those early years, because I think people outside molecular biology sometimes imagine a discovery like this arriving in a single dramatic instant, when in reality it arrived through hundreds of hours of tedious, repetitive bench work that happened to accumulate, sample by sample, into something extraordinary. Every hair sample had to be cleaned of surface contaminants before extraction, using a wash protocol involving several rinses in a detergent and ethanol solution to remove any DNA that might have been deposited on the outside of the shaft by handling or by contact with other organic material in the field. The follicle, if one was present, was cut away from the shaft and processed separately, since follicular tissue yields nuclear DNA in addition to the mitochondrial DNA present in the hair shaft itself, though for most of the project's early years I worked almost exclusively with mitochondrial markers, both because they are more abundant and easier to amplify from degraded field samples and because the existing reference databases for species identification were built primarily around mitochondrial sequences, making comparison more straightforward. I ran polymerase chain reaction amplification using primers targeted at conserved regions flanking the more variable cytochrome B gene, a standard approach in wildlife forensics that allows a small quantity of degraded, field-collected DNA to be copied millions of times over, producing enough material to sequence reliably. Each run took the better part of a day, from extraction through amplification, through gel electrophoresis to confirm a successful amplification before sending the product out for sequencing. And in the early years, before the university's own sequencing capacity had expanded, I was sending samples to an outside contract lab in Vancouver and waiting 2 to 3 weeks for results, which meant that the pace of discovery for most of 1997 and 1998 was agonizingly slow. A handful of confirmed results trickling in every few months rather than the steady stream of data a better funded project might have generated. I mention this not to complain about resource limitations which every field scientist learns to work within, but because I think it matters for understanding why this project took 15 years rather than 5. I was doing all of this alongside a full teaching load, alongside my funded caribou and salmon work, alongside serving on departmental committees and supervising other graduate students whose projects had nothing to do with any of this, and alongside, increasingly through those years, trying to be present for Joan in a marriage that was, for long stretches, absorbing far more of my attention and energy in secret than either of us had originally agreed to when we decided together back in 1995 that I would take the position in Prince George. I want to tell you about one particular stretch in the winter of 1999 into 2000 that illustrates both the isolation of that work and the toll it was quietly taking. Raymond had brought me a third sample in November of that year, a set of hair caught on a fence post at one of his cabin sites, and I had spent most of December running the extraction and amplification during evenings after Joan had gone to bed, telling her only that I had a backlog of grant-funded samples I needed to clear before the holidays, which was true of my daytime work, but not of what I was actually doing at 11 o'clock at night in a lab I had to badge into after hours. The sample confirmed the same lineage, a result I received by fax from the Vancouver contract lab on the 23rd of December, and I remember sitting alone in my office that evening, the building nearly empty for the holiday break, reading the sequence alignment against my growing reference set and feeling a combination of scientific excitement and a much more uncomfortable loneliness because I had no one in that building, no colleague down the hall, no departmental seminar in whom I could confide what I was actually looking at. I drove home that night and told Joan I had gotten good news on a grant sample, which again was not exactly a lie, and I remember her asking with the particular perceptiveness she has always had why good news seemed to be making me look so tired rather than pleased. I did not tell her the full truth for another 3 years. I have never fully forgiven myself for that gap, even though I understand, looking back, why I made the choice I made at the time. The other thing I want to describe from those early years is the sheer physical difficulty of the fieldwork itself, because I think it is easy, listening to a summary account like this one, to underestimate how much of the project's credibility rested on the fact that Raymond and I were doing genuinely demanding backcountry travel year after year in conditions that would deter almost anyone without decades of experience in that specific terrain. The Telkwa Range is not a place with maintained trails above the tree line. Access into Hunter Basin required either a long approach on foot from the nearest logging spur, gaining nearly 4,000 feet of elevation over roughly 14 kilometers, or in later years when I could occasionally justify the expense against legitimate grant's field logistics budget, a helicopter charter out of Smithers that could put us into a rough landing zone below the basin in under 20 minutes, but that came with its own risks, given how quickly weather can close in on that country even in midsummer. I turned 50 in 1999, and by 2002, when I saw my first track with my own eyes rather than through Raymond's photographs, I was beginning to feel, in my knees and my lower back after each trip, the cumulative cost of nearly a decade of high-altitude field seasons layered on top of the ordinary demands of the job I was actually being paid to do. Raymond, 12 years my senior, never once complained about the terrain in front of me, though I suspect now, knowing him as well as I eventually came to, that he managed his own physical limits with a stoicism that occasionally cost him more than he ever let on. I want to spend a moment on the man who was quietly feeding this entire project, because Raymond Joseph deserves more credit than the role of anonymous sample courier. Raymond had grown up on his family's trapline inherited from his father in a stretch of the Telkwa Range that his family had worked for generations before the province formalized the registered trapline system in the 1920s. He knew that country better than any biologist, myself included, would ever know it from a handful of summer field seasons. He was skeptical of outside institutions in the reasonable way that many people in rural and indigenous communities in that part of the province had learned to be skeptical of universities and government agencies that had a long history of extracting knowledge and resources without much in the way of reciprocity or respect. He trusted me as far as I could tell, because I did exactly what I said I would do with his samples and never once tried to publicize his name or his trapline location without permission. That trust took years to build, and I never took it for granted. That trust took years to build, and I never took it for granted. I want to describe a conversation I had with Raymond in the summer of 2001, because it changed how I understood the entire project, and because I think it belongs in this account, even though it produced no data, no sample, nothing that would appear in any of my notebooks. We were sitting outside his cabin on a warm evening in July, waiting on a pot of coffee heating over his camp stove, and I asked him as directly as I ever had up to that point how long his own family had known about what we were investigating. He was quiet for a while, the way he often was before answering a question that mattered to him, and then he told me that his grandmother, who had lived to be well past 90 and had passed when Raymond was a young man in his 20s, used to tell him and his cousins stories about beings who lived high in the mountains above the valley, beings who kept to themselves, who were understood by his family and by other Wet'suwet'en families in that country to be neither wholly separate from people, nor the same as people, occupying a category of being that Raymond said did not translate cleanly into English, though the closest he could come, in the language we were both using that evening, was something like, "The tall ones who watch from a distance and do not interfere unless interfered with first." He told me that his grandmother had described specific protocols passed down through generations for how a person should behave if they came across sign of these beings while out on the land, how to leave food without approaching too closely, how to avoid direct, prolonged eye contact if an encounter happened at close range, how certain places in the mountains were understood, without anyone needing to post a sign or draw a map, to belong more to them than to any human family's trapline or hunting ground, regardless of what the provincial registered trapline system said on paper. He said his grandmother had never described these beings with fear in her voice. Not the kind of fear you would use for something dangerous or malevolent, but with a specific kind of respect that Raymond said he had come, over his own decades in that country, to understand from direct experience rather than simply from inherited story. I asked him that evening why he had decided to bring the hair sample to a university lab at all, if his own family's knowledge and protocols had already told him everything he needed to know. He thought about that question longer than any other I had asked him up to that point. And when he answered, he said something I have carried with me for the rest of this project, and I suspect for the rest of my life. He said that his grandmother's knowledge had never needed a university's confirmation to be true, and that he was not looking for a scientist to tell him something was real that he had never doubted. What he wanted, he said, was a documented record that would exist outside of oral tradition alone, something that could stand up in the kind of institutional spaces, land use hearings, environmental review processes, resource development consultations, where oral tradition was too often dismissed or discounted by exactly the kinds of officials I would later find myself negotiating with over location data. He wanted a second kind of evidence built in a different language than the one his grandmother had used, that could sit alongside what his family had always known and make it harder for anyone with the authority to approve a mine or a pipeline through that country to pretend the question had never been properly investigated. That conversation reframed the entire project for me in ways that took years to fully settle. I had been thinking of myself up to that point as the person discovering something. After that evening, I understood myself more accurately as a person documenting in one particular institutional language something that had already been known carefully and precisely by people who had lived alongside it for longer than any university had existed to grant or withhold credentials for the privilege of confirming it. That understanding is, I think, the reason I was ultimately willing to walk away from my career rather than hand precise coordinates to a government database, because doing so would not only have endangered a population of previously undocumented hominids, it would have betrayed a specific promise made to a specific man on a specific summer evening that the scientific record I was building would serve to protect what his grandmother's protocols had protected for generations, not become the instrument that finally exposed it to exactly the kind of institutional overreach those protocols had always been designed to guard against. By 2002, 6 years into the project, I had accumulated enough data that the scientific question had shifted from what species is this to something closer to how large is this population and what is its taxonomic relationship to known hominid lineages. And I want to be direct about the fact that by this point I could no longer avoid that word in my own private thinking, even if it never appeared in anything I wrote for institutional consumption. The mitochondrial sequences I had recovered showed a closer relationship to the broader hominid clade than to any bear, wolf, or ungulate lineage, though the divergence from documented hominid sequences, including human, chimpanzee, gorilla, and orangutan reference sequences, was substantial enough that this was clearly not a hybrid or contamination artifact involving any known hominid lineage. It was, as best I could determine using the phylogenetic software available to me at the time, a lineage that had diverged from the broader hominid family tree at a depth roughly comparable to, though distinct from, the divergence separating gorillas from the chimpanzee human lineage. In plain terms, whatever was leaving hair and scat in the Telkwa range was more closely related to us than any bear is, but it was not any documented hominid lineage, and it was certainly not human. I ran that same phylogenetic comparison against every hominid reference sequence I could find, including several less commonly cited datasets from smaller regional research collections that most labs would not have thought to check, partly out of thoroughness and partly, I will admit, out of a lingering hope that some overlooked entry would resolve the anomaly into something ordinary. None of them narrowed the gap. The lineage remained exactly where the earlier analysis had placed it, distinct enough from every documented branch of our shared family tree that no amount of additional reference data was going to make the finding disappear, which is, I think, the moment a careful scientist has to stop looking for the mistake and start treating the result as real. I need to tell you about the first time I saw a track myself rather than relying on Raymond's photographs and measurements, because it marked a shift in how seriously I was taking my own physical safety in pursuing this research. In August of 2002, Raymond invited me to accompany him on a 2-day trip into the Upper Basin, ostensibly to check on a research camera I had asked him to help me place the previous summer. A basic infrared trail camera model that was, by the standards of 2002, reasonably sophisticated but still limited by film capacity and battery life compared to what would become available a decade later. We hiked in from a logging spur road that dead-ended below Hunter Basin, climbing through subalpine fir and mountain hemlock before breaking into open alpine meadow scattered with heather and stunted whitebark pine, the kind of high, wind-scoured country where snow can persist into July even in a mild year. We found the track on the second morning in a patch of soft mud at the edge of a tarn, a small alpine lake fed by snowmelt at an elevation I estimated using my handheld GPS at just over 5,500 feet. The print was fresh, made sometime in the previous 12 hours based on how crisp the edges were and the fact that no overnight frost had blurred the impression, which told me it had been made after the temperature rose above freezing that morning, meaning within a few hours of when we found it. It measured 15.5 inches long by roughly 7 inches wide at the ball of the foot, with a discernible arch and 5 toes, the big toe notably larger and more separated from the others than a human foot structure would produce, though the overall gait pattern, based on the stride length to the next print roughly 5 feet away, was clearly bipedal and clearly not a quadrupedal animal's track pattern, misread by an inexperienced observer, which is the most common explanation offered by skeptics for reported tracks of this kind. I photographed the print from multiple angles with a scale bar, made a plaster cast using a kit I had brought specifically for that purpose, and recorded the GPS coordinates, elevation, substrate type, and estimated age of the print in my field notebook, with the same rigor I would apply to documenting a grizzly track for a ministry survey. I want to emphasize this because it matters for what happened later. I treated this evidence exactly the way I would treat any piece of field data in a legitimate wildlife study, because I had, by 2002, become convinced that whatever this population was, it deserved the same standard of documentation as any other undescribed species, and sloppy, sensationalized evidence would only ever set back the eventual scientific reckoning that this discovery, if it held up, would require. We recovered the trail camera that same afternoon. It had been in place for 11 months, mounted on a lodgepole pine at the edge of a game trail that Raymond had identified as heavily used based on years of observation. The camera had captured 211 frames over that period, the vast majority of them triggered by wind-moved branches. A mule deer, 2 black bears at different times of year, and a pine marten that had apparently investigated the camera, housing itself, judging by the extreme close-up frames of what was clearly marten fur pressed against the lens. Nothing anomalous. I want to be honest about that, because it would have been easy, given everything else that had accumulated by that point, to overstate the significance of an ambiguous frame if one had existed. None did. The camera simply had not captured what we were looking for, which is itself an unremarkable and expected outcome given how large the search area was relative to the coverage of a single fixed camera. I am telling you about this trip specifically because of what happened on the hike back out on the evening of the second day, roughly an hour before we reached camp. Raymond stopped abruptly on the trail ahead of me, raised one hand, and stood motionless for perhaps 10 seconds before crouching slowly and gesturing for me to do the same. I did not see anything at first. We were in a band of subalpine fir with enough undergrowth that visibility beyond 30 or 40 meters was limited. Then I heard it. A vocalization, low in pitch, sustained for perhaps 3 seconds with a quality that I can only describe as a deep chest tone, the kind that suggested a chest cavity considerably larger than a human's, coming from somewhere below us and to the west, perhaps 200 meters away based on how the sound carried through the timber. It was answered, after a gap of maybe 15 seconds, by a second vocalization from a different direction, higher in pitch, shorter in duration from somewhere above us on the slope. I did not see anything that evening. I want to be clear about that because I am not going to inflate this account with a sighting that did not happen. What I experienced was auditory, and while auditory evidence is inherently less rigorous than a visual sighting or a physical sample, I had, by 2002, spent enough years in that country and reviewed enough bioacoustic literature on North American wildlife vocalizations to say with professional confidence that what I heard did not match any documented call of an elk, wolf, owl, or any other species native to that ecosystem. Raymond, for his part, said only, "That's them," in a tone that suggested he had heard this exact exchange many times before and had simply been waiting to see whether I would hear it too. I did not have a full visual encounter until 3 years later in August of 2005, and I have relived that afternoon often enough in the years since that I can still walk through it minute by minute in a way I cannot say about most days from that period of my life. Raymond and I had gone in to service the second trail camera, a newer digital unit I had purchased that spring with money from my own savings rather than any grant, mounted at a pinch point along a game trail below Hunter Basin, where 3 separate drainages converged into a single narrow creek crossing, a location Raymond had selected specifically because it funneled animal movement through a corridor no more than 15 meters wide between a rock outcrop and a stand of dense subalpine fir. We had hiked in that morning under a clear sky, the kind of high blue August day that the coast mountains and the interior ranges rarely produce more than a handful of times each summer, cold enough at 5,000 feet that we still had frost on the tent fly when we broke camp, warm enough by midday that I had stripped down to a base layer while we climbed the final switchbacks into the basin. We reached the camera station a little after 2 in the afternoon. Raymond stopped ahead of me the way he always stopped, with a stillness that I had learned over 9 years to read as more informative than any spoken warning, and this time he did not crouch. He stood exactly where he was, facing across the creek toward the rock outcrop, and he said, very quietly without turning his head, "Wallace, don't move yet, just look." It was standing at the base of the outcrop, perhaps 35 meters from where we stood, partially shaded by an overhang of gray lichen-covered rock, but caught in the upper portion of its body in a shaft of direct afternoon light that made the moment feel less like a wildlife sighting and more like something staged, though nothing about it was staged. It was upright on 2 legs, with a posture that was unmistakably bipedal rather than an animal, balanced momentarily on its hind legs, which is the explanation skeptics reach for most often and which collapses immediately once you have actually seen the thing itself, because there is a quality to sustained bipedal stance, a distribution of weight through the hips and the arch of the foot that no bear or ungulate can hold for more than a few seconds, and this figure had clearly been standing that way before we ever came into view. Unhurried, its weight settled evenly, one long arm resting against the rock face beside it. I am going to try to describe what I saw with the same precision I would use to describe any piece of field data, because that discipline is the only thing that kept me functioning in that moment, rather than simply staring in a kind of paralysis that would have cost me the details I spent the following hour writing into my field notebook before the memory could soften into something less exact. The body was covered in hair, dark brown shading toward black across the shoulders and back, with a slightly lighter, almost reddish cast along the forearms where the afternoon light caught it directly. The hair was not uniform in length. It was noticeably longer and shaggier across the crown of the head and down the back of the neck, shorter and closer to the body across the chest and upper arms, a pattern I would later find echoed on a much smaller scale in the microscopic cross-sections I had already been running on the hair samples for years, though seeing it distributed across a living body made a set of numbers in a lab notebook suddenly viscerally real in a way that no data table ever could. I estimated its height at somewhere between 7.5 and 8 feet, a judgment I made by comparing its shoulder height against the rock outcrop it stood beside, a formation Raymond and I had both measured with a laser rangefinder 2 years earlier for an unrelated snow depth study, so I had an unusually precise reference point available to me in that instant, one I would not normally have in the field. Its shoulders were broad relative to its height in a way that no human body, however large, achieves, and its arms hung with a length that brought its hands to a point well below the knee, hands that I could see clearly enough, even at 35 meters, to note were proportioned differently from a human hand, with a thumb that appeared set lower and more separated from the fingers than our own opposable thumb sits. The face is the part I have been asked about more than any other detail in the rare conversations I have had about this sighting over the years, and it is the part I find hardest to render into language that does the actual experience justice. It was not a human face, and it was not an animal face in the sense that a bear's face or a deer's face reads immediately and completely as animal, without any of the ambiguity that makes you look twice. It occupied a middle register that I think is precisely why encounters like this one produce such a visceral reaction in the people who have them, because our visual processing, built over a lifetime of sorting faces into human and not human almost instantaneously, does not have a comfortable category for a face with a heavy, pronounced brow ridge, a broad, flattened nose, and a jaw structure wider and more forward than our own, arranged nonetheless with an expression in the set of the mouth and the angle of the head that read unmistakably as attention rather than as blank animal wariness. It was looking at us, not glancing, not startled and assessing an escape route the way a bear does in the half second before it bolts. Looking with a steadiness that I recognized uncomfortably as the same quality of directed regard I would use myself when observing something I found genuinely interesting. We stood like that for what my field notes, written immediately afterward, estimate as somewhere between 40 and 50 seconds, though in the moment it felt considerably longer, stretched the way time stretches during any event, significant enough to override the ordinary sense of duration. Raymond did not move. I did not move, in part because Raymond's stillness had trained me over 9 years of field work together to trust his read of a situation over my own instincts, and in part because some deeper, less analytical part of my own reaction simply did not want to break whatever fragile equilibrium was allowing this to continue. It broke first, not with any sudden motion, not with the explosive flight response you would expect from a startled animal, but with a single, unhurried step backward into the deeper shadow at the base of the outcrop, followed by a low vocalization, shorter and softer than the exchanges I had heard from a distance 3 years earlier that I felt as much as heard, a resonance that seemed to move through the rock face itself before it reached my ears. Then it was gone, not with any sound of retreating footfalls or breaking branches, simply gone, the space where it had been standing returning to being an unremarkable patch of shadowed rock and lichen, as though nothing had ever occupied it. Raymond let out a breath I had not realized he was holding. He looked at me and said, "Now you've seen one." It was not a question. I did not answer it as one. I sat down on the nearest flat rock, took out my field notebook, and spent the next hour writing down every detail I could recall in the order I recalled it before we said another word to each other about what we had just witnessed, because I understood, even in that state, that this was the single most important piece of data I would ever collect in this project, and that the accuracy of my own memory, more than any sample or any sequence, was the instrument I needed to protect. In that moment, I did not photograph it. I want to be honest about that because I know it is the detail that any skeptical viewer will fix on immediately. My camera was in my pack, slung across my back, and by the time I had processed what I was looking at clearly enough to think about retrieving it, I understood, with the same instinct that had kept me still through the entire encounter, that reaching for equipment would end the moment immediately, and that whatever this population was, it had allowed itself to be seen, briefly and at a careful distance, in a way that felt intentional rather than accidental. And I was not willing to trade that for a photograph I was not even confident I could have captured in time. I have thought about that decision many times in the years since, weighted against what a photograph might have meant for the credibility of everything that came after, and I have never fully resolved whether it was the right choice. I only know it was the choice I made in the moment without deliberation, and that Raymond, when I asked him about it afterward, told me he would have made the same one. Before I go further into what the following decade brought, I want to pause on something practical, because I know some of you watching are wondering how any of this survived institutional scrutiny at a university that depended on government and industry funding for its credibility. The answer is that for the first several years, it did not survive institutional scrutiny because it was never presented for institutional scrutiny. I kept this work almost entirely separate from my funded research portfolio. I published during this same period 4 peer-reviewed papers on caribou and salmon genetics that had nothing to do with any of this, and those papers were what my department head, my funding agencies, and my tenure committee saw and evaluated. The Telkwa project existed in a parallel track, funded out of my own pocket for equipment costs beyond what I could quietly absorb into existing grants, documented in a set of notebooks I kept at home rather than in my university office, and known to almost no one except Raymond and eventually my wife. Joan and I had married in 1990, 3 years before I moved north for the UNBC position, and she had followed me to Prince George with the kind of patient good humor that a marriage to a field scientist requires. She taught high school chemistry, which meant she understood the difference between rigorous evidence and speculation as well as I did. And when I finally told her in the winter of 2003 roughly what I had been doing on my supposed solo camping trips for the previous several years, her first response was not disbelief. Her first response was to ask whether I had controlled for contamination in my extraction protocol, which is exactly the question I would expect from a scientist's spouse and exactly the reason our marriage survived what came later. She did not believe me immediately. She wanted to see the sequence alignments herself, which I showed her, spread out across our kitchen table on a Saturday afternoon in January, and I watched her go quiet in the particular way that a rigorous person goes quiet when the data in front of them will not resolve into any comfortable, conventional explanation. From 2003 onward, Joan became, in effect, an unofficial and unpaid research partner. She helped me maintain the notebooks. She read early drafts of the analysis I was slowly building toward an eventual formal presentation. And she was the one who, in the spring of 2004, suggested that if I was going to keep doing this work seriously, I needed a second, truly independent source of samples from outside the Telkwa drainage, because a pattern confirmed in only one location, however many samples I collected there, was vulnerable to the criticism that I was simply looking at a single, unusual population, a genetic curiosity confined to one isolated valley rather than evidence of a broader, previously undocumented species with a wider range. That search for a second location took nearly 2 years and ultimately succeeded through a connection I would never have anticipated. In the fall of 2005, I gave a guest lecture at the University of Northern British Columbia's Prince George campus on wildlife DNA forensics, a routine presentation aimed at undergraduate students considering graduate work in molecular ecology and afterward a ministry of forests. Biologist named Colin Presley approached me with a question about hair sample degradation rates in cold storage. Colin worked out of the ministry's Bulkley-Nechako district office and spent much of his fieldwork season doing fur-bearer population surveys across the district that stretched from the Bulkley Valley north into the Babine Range and west toward the Nass watershed near the town of New Ayanch. Over coffee after the lecture, in the kind of casual professional conversation that often produces more useful leads than any formal research proposal, I mentioned, carefully, that I was interested in unusual hair samples from remote high-elevation terrain, framing it as a general biodiversity interest without specifying what I actually suspected. Colin mentioned that he had, 2 years earlier, collected a hair sample from a hair snag station he had set up for a wolverine survey in the Babine Range, north of Smithers, that had never been successfully identified by the standard lab his ministry district used at the time, a private contract laboratory in Victoria that ran a more limited reference database than my own university lab did. He said the sample had simply been logged as unidentified and filed, since a single unmatched sample out of hundreds collected across a multi-year wolverine study was not, on its own, worth pursuing further within the scope of that project's funding and mandate. He sent me the sample the following week. It's sequenced to the same mitochondrial lineage as the Telqua samples, with a level of divergence between the two populations consistent with genetic separation across a distance of roughly 140 kilometers of intervening mountainous terrain. The kind of divergence you would expect between two populations of the same species that had limited but not entirely absent gene flow between them, likely maintained by occasional long-distance dispersal of individuals across connecting high-elevation corridors, a dispersal pattern well documented in other large-bodied mountain mammals like wolverine and grizzly bear in that same landscape. Colin and I made 2 joint field trips into the Babine Range over the following 2 years in the summers of 2006 and 2007, using his existing Wolverine survey infrastructure as cover for the additional sampling I wanted to do. A practical arrangement that also happened to be entirely legitimate, since hair snag stations built for wolverine census work catch the hair of anything large enough to brush against the barbed wire, and there was no reason Colin could not simply expand his own routine data collection to include a wider set of species while he was already out there doing the survey work his actual job required. The Babine Range trips confirmed something I had not fully appreciated from the Telqua work alone, which was how much the local topography shaped where evidence concentrated. In the Telqua drainage, Raymond's decades of trapline experience had already narrowed the productive search area to a specific 60 square kilometer basin. In the Babine Range, Colin and I had no equivalent local knowledge to draw on, and our first season in 2006 produced almost nothing beyond the original sample Colin had collected 2 years earlier, a frustrating stretch of 10 days spent checking snag stations across a much wider, less targeted area with only marginal results. It was Colin himself, reviewing his own wolverine movement data that winter, who noticed a pattern worth testing. The wolverine detections across his survey grid clustered disproportionately around a specific set of high alpine cirques on the range's eastern flank. Terrain that offered the kind of year-round access to marmot and ground squirrel prey that would support wolverine territories through the winter, and Colin reasoned, correctly as it turned out, that any species relying on similar prey resources and similar terrain characteristics for winter survival might show the same clustering pattern. We shifted our 2007 effort entirely to those cirques and recovered 3 additional samples in 8 days, a hit rate high enough that I began for the first time to think seriously about habitat modeling as a tool for this project rather than simply relying on opportunistic sample collection and Raymond's decades of accumulated local knowledge. I never had the chance to build that modeling work out properly before the university dispute consumed most of my remaining research time, and it remains, alongside the precise population size question, one of the open scientific questions I think about most often in retirement, one that a better resourced, longer-running project than mine would eventually need to address properly. This was the finding that, more than any single piece of evidence before it, convinced me I was looking at something with a genuine, if narrow, geographic range across the Skeena and Nechako watersheds, rather than an isolated genetic curiosity in one valley. And it was also, I want to be honest, the finding that made me start seriously considering for the first time what I owed to the broader scientific community in terms of disclosure, and what the consequences of that disclosure would likely be. I brought Colin partially into confidence at that point, though I still did not use the word Sasquatch with him directly. I described the project as investigating an undocumented, possibly relict hominid lineage in the region, phrased in the driest possible scientific register, and I watched him process that phrase with the particular stillness of a field biologist who had spent enough years in that country to have his own private suspicions that he had never voiced to a university researcher before. He agreed to continue forwarding any unusual samples from his ongoing fur-bearer and carnivore survey work, and over the following 4 years, from 2005 through 2009, he sent me 11 additional samples, 9 of which sequenced to the same lineage, expanding my documented range to include portions of the Babine Range, and, in 2008, a single hair sample recovered from a cut block edge near the Nass watershed that pushed the documented range more than 200 kilometers from the original Telkwa population. I need to explain what I was doing with all of this data during these years because I was not simply accumulating samples for their own sake. I was building, slowly and as rigorously as I knew how, toward a mitochondrial phylogeny robust enough to establish beyond reasonable methodological doubt that this lineage represented a genetically distinct, previously undescribed hominid population with an estimated combined range across at least 3 major watersheds in north-central British Columbia. I ran full mitochondrial genome sequencing rather than just the cytochrome B fragment on the 4 highest quality samples in 2007 using capacity I was able to access through a collaborative arrangement with a genomics facility at the University of British Columbia that I justified to the facility director as a side project related to carnivore phylogenetics, which was, again, not technically a lie, simply an incomplete truth. The full mitochondrial genome data confirmed and sharpened what the shorter cytochrome B fragments had already suggested. The lineage's divergence time from the common ancestor it shared with modern humans and other hominids, calculated using standard molecular clock methods calibrated against known hominid fossil divergence dates, placed the split at somewhere between 400,000 and 700,000 years before present, with a wide confidence interval reflecting the inherent uncertainty of molecular clock estimates, but a range that placed this lineage's divergence roughly contemporaneous with, though distinct from, the divergence of Neanderthals and Denisovans from the modern human lineage in Eurasia. This was not a population of unusually large or unusually hairy humans. This was not a surviving population of any documented extinct hominin species from the paleoanthropological record, none of which had ever been associated with North America prior to the arrival of anatomically modern humans across Beringia. This was something else, a separate, undocumented hominid lineage present in North America with a genetic signature that did not correspond to any migration event or population currently recognized in the paleoanthropological or genetic literature covering the peopling of the Americas. I sat with that finding for the better part of a year before I told anyone beyond Joan, Raymond, and Colin. I want to try to convey what that year was like because I do not think most people, even most scientists, fully appreciate what it means to hold a finding of that magnitude in isolation without the normal scaffolding of peer collaboration, conference presentation, and gradual community vetting that most significant scientific discoveries pass through. I would sit in my home office at night after Joan had gone to bed, running the phylogenetic trees again and again, checking my assumptions, looking for the error that would make all of this go away and let me return to caribou genetics and salmon population studies in a career that made sense within the ordinary structures of academic life. I never found that error. Every time I reran the analysis, using different software packages, different reference sequences, different calibration points for the molecular clock, the result held. Somewhere in the mountains of north-central British Columbia, a population of hominids, genetically distinct from every human population on Earth, had been living and reproducing for hundreds of thousands of years, entirely unknown to Western science, and I was apparently the first person to have gathered enough physical evidence to prove it with the kind of rigor that could withstand peer review. The question that occupied that entire year was not whether the science was sound. By 2008, I was confident the science was sound, confident enough that I began drafting in secret a manuscript intended for submission to a major journal, structured exactly as I would structure any species description paper, with full methods, full phylogenetic analysis, and a discussion section that I wrote and rewrote perhaps 30 times, trying to find language that would present an extraordinary conclusion with the same measured, defensible tone I would use to describe a new subspecies of vole. The question that occupied that year was what publication would actually mean for the population itself, for Raymond and Colin, who had trusted me with information about specific locations, and for my own career and my family's stability. I discussed this at length with Joan across many evenings at our kitchen table, and I discussed it more cautiously with Raymond, whose perspective mattered more to me than almost anyone else's because he was the person whose trust had made this entire project possible and whose relationship to that landscape long predated my own by generations. Raymond's position when I finally asked him directly what he thought I should do with 15 years of accumulated evidence was more complicated than I expected. He was not opposed to the science being documented. He told me in a conversation I still remember almost word for word that his own family and community had known about these beings for longer than there had been a Canada or a British Columbia to draw a border around them, and that Western science arriving to confirm what his grandparents had always known was, in his words, about 100 years late and asking the wrong people for permission. But he was deeply opposed to any publication that would specify locations precisely enough to draw hunters, thrill seekers, or worse, government agencies interested in resource extraction or land use decisions into country that had, up to that point, remained protected largely by its remoteness and by the discretion of the small number of people who knew what was actually out there. There is one more relationship from those middle years I need to describe before I move into what happened once the university took control of the project, because it illustrates the same tension in a smaller, more personal register, and because it still weighs on me more than almost anything else from that period. In the fall of 2007, a graduate student named Trevor Adsett joined my lab to begin a master's thesis on carnivore population genetics along the Highway 16 corridor, funded through a legitimate grant I held studying wolf and cougar territorial overlap in areas of increasing human-wildlife conflict near expanding forestry operations. Trevor was bright, meticulous, and within his first several months in the lab, sharp enough to notice that a portion of my freezer space and a recurring, unlabeled category of samples in my private notebook did not correspond to anything in his own project or in any of the other funded work moving through the lab that year. He asked me about it directly in the way that good graduate students eventually learn to ask their supervisors direct questions in March of 2008, and I made the decision, one I debated for weeks beforehand, to bring him partially into the project, primarily because I needed a second set of hands I trusted for some of the more technically demanding full genome sequencing work, and because Trevor had by that point proven himself exactly the kind of careful, unshowy scientist who would treat an extraordinary claim with appropriate rigor rather than excitement. I did not tell him everything at first. I told him I was investigating an unusual, potentially undescribed carnivore or hominid lineage from samples collected in the Bulkley Nechako region, and I asked him to help me run additional phylogenetic comparisons without telling him what species I suspected he was looking at. A blind analysis design that, in retrospect, was as much about protecting myself from confirmation bias as it was about protecting the project's confidentiality. Trevor ran the comparisons in April of 2008 and came into my office 2 days later with a printed phylogenetic tree in his hand and an expression I had never seen on him before, somewhere between excitement and something closer to unease. He asked me, without any preamble, whether I understood what the divergence date on this tree implied. I told him I did. He asked why, if that was true, we were not treating this as the most significant finding either of us would likely ever be associated with in our careers. I told him as carefully as I could about Raymond, about the trap line, about the 15 years, at that point 11 years of careful accumulation that had gotten me to this exact tree, and about my conviction that the value of this discovery would be measured not in how quickly it was announced, but in whether it survived contact with the institutions that would eventually have to evaluate it. Trevor stayed with the project for 2 more years, through the end of his master's degree in the spring of 2010, and he became, alongside Joan and Raymond, one of the very small number of people who understood the full scope of what I had built. He was also, I want to be honest, the person whose growing frustration with my caution most directly foreshadowed the pressure that would eventually come from the university itself. Trevor was 26 years old in 2009, ambitious in the ordinary, healthy way that good graduate students are ambitious, and he did not share my instinct toward indefinite restraint. He argued with me more than once that a discovery of this magnitude belonged to science as a whole, not to the 2 of us and a retired trapper, and that every year we delayed formal publication was a year in which some other researcher working from different samples in a different drainage might beat us to a discovery that 15 years of my own life had built. He was not wrong about the risk, and I told him so. I was simply not willing to accept that risk if the price of avoiding it was precise location data in an unrestricted institutional record. The disagreement came to a head in January of 2010. During the same winter, the university's pressure was building toward the meeting that would eventually cost me my career, when Trevor, without asking my permission, drafted his own version of a manuscript using the phylogenetic data he had helped generate, intending to submit it independently as a co-authored paper with a location precision that went well beyond what I had ever agreed to disclose, on the grounds, as he explained to me when I discovered the draft on a shared lab computer, that the scientific community deserved reproducible location data and that my restrictions amounted to unscientific gatekeeping of a discovery that was no longer solely mine to control. I want to be fair to Trevor even now because I understand his position better than I did at the time, and because the argument he made was not a foolish one. Reproducibility is a legitimate cornerstone of scientific method, and a paper that cannot be independently verified by other researchers working from the same locations carries real epistemic weaknesses that a purist would be right to point out. But I also understood, in a way that a 26-year-old graduate student building his career had no personal stake in understanding, exactly what precise location data would mean for Raymond's trapline, for the handful of family groups I had spent over a decade documenting without ever seeing them harassed, hunted, or displaced by the kind of attention that precise coordinates would inevitably bring, regardless of what any journal's stated ethics policy claimed to protect. I confronted Trevor about the draft in February of 2010. It was not a comfortable conversation, and it ended, ultimately, with Trevor withdrawing the independent submission at my direct request, but also with a rupture in our working relationship that never fully healed. He finished his degree that spring, moved to a doctoral program at the University of Victoria, studying an entirely unrelated question in coastal marine genetics, and we have exchanged perhaps 4 emails in the 14 years since. I do not blame him for his ambition. I have wondered more than once in the years since my own career ended whether his instinct to move faster and disclose more broadly might have produced a better outcome than my own caution did, whether a paper with full location data published in 2010 under joint authorship with the university's institutional backing might have generated the kind of conservation protections that watershed-level data published independently in 2012 under my name alone never came close to achieving. I do not know the answer to that question, and I have made my peace with not knowing it, but I think about Trevor Adsit more often than almost anyone else from this entire story, because he is the person who forced me earliest and most directly to confront the fact that my own caution had costs too, not just benefits, and that there was no version of this project that would have let everyone involved end up satisfied with the outcome. He is the person who forced me earliest and most directly to confront the fact that my own caution had costs too, not just benefits, and that there was no version of this project that would have let everyone involved end up satisfied with the outcome. The last of those 4 emails came in the fall of 2021 after Trevor, by then an associate professor himself at a university on the coast, apparently came across a citation to my 2012 paper while researching an entirely unrelated question about mitochondrial divergence dating methods. He wrote to tell me briefly and without much elaboration that he had thought about our disagreement many times over the intervening decade, that his own experience navigating academic politics as a junior faculty member had given him a considerably more sympathetic view of the caution I had insisted on back when he was 26, and certain that speed and full disclosure were the only responsible path forward. He did not apologize exactly, and I did not expect him to, since I have never believed he owed me one. But he closed the email by saying that he hoped, if either of us ever found ourselves in a position to collaborate again, that we would take it, and I wrote back that I hoped so too, though neither of us has yet found the occasion, and I suspect, given where both our careers now stand, we probably never will. I keep that email printed in the same folder as Marcus Fielding's letter because together they represent the 2 people from the university years whose eventual hard-won understanding of what I chose to protect has mattered to me more than any recognition the broader scientific community was ever going to offer. That tension, between the scientific imperative to document precisely and reproducibly, and the ethical imperative to protect a population that had survived this long, specifically because no one had ever pinned it to a map with enough precision to bring in the kind of attention that ends populations, is the tension that ultimately defined the rest of my career. And it is the tension that eventually ended it. In January of 2009, I made a decision that I have gone back and forth on for years since, and that I still am not entirely certain was the right one, though I have made my peace with it. I decided to bring my department head, a wildlife biologist named Dr. Sandra Whitlock, into partial confidence, not because I had resolved the tension between disclosure and protection, but because I had reached the limits of what I could responsibly do as a solo researcher working outside institutional channels, and because I believed, perhaps naively, that bringing the work under formal university oversight would allow me to pursue proper peer review while retaining some control over how location data was handled. Sandra had been my department head for 4 years by that point, and I respected her as a rigorous scientist, and I believed at the time as someone who would treat an extraordinary claim with the appropriate combination of skepticism and open-mindedness. I scheduled a meeting, brought a summary of the mitochondrial data, the phylogenetic analysis, and photographs of the physical evidence I had collected over 13 years, and I laid it out for her exactly as I would present any research finding, methodically, with full acknowledgement of the limitations and uncertainties in the data. She was quiet for a long time after I finished, longer than I expected. Then she asked me the question I had been asking myself for a year. Who else knows about this? I told her. She asked whether I had submitted anything for publication. I told her I had a draft manuscript but had not submitted it anywhere. She asked me to give her a week before we discussed next steps. I do not know exactly what happened during that week, though I have pieced together a partial picture from conversations in the years since with people who were present for parts of it. What I know is that by the end of that week, the situation had moved well beyond my control. Sandra had, apparently believing she was following appropriate institutional protocol for a finding with potentially significant implications, briefed the university's vice president of research, who in turn briefed someone at the provincial Ministry of Environment, on the grounds that a discovery of this magnitude, if confirmed, would have regulatory and land management implications that fell within the ministry's mandate, given that any newly documented species would trigger conservation status assessments and potential habitat protection requirements under provincial and federal wildlife legislation. Within 3 weeks, I was no longer the sole custodian of 15 years of careful, quiet work. I was one voice in a rapidly expanding circle of university administrators, government officials, and eventually outside experts brought in to evaluate my findings, most of whom had no background in the specific methodological questions involved, and many of whom I came to believe were far more interested in the institutional and political implications of the discovery than in the underlying science. The next 2 years, from early 2009 through the spring of 2011, were the most difficult period of my professional life, and I want to walk through them carefully because this is the part of the story that most directly explains how my career ended, and I think the details matter more than a simple summary would convey. The university's initial response once the finding had moved up the administrative chain was surprisingly cautious and, for a brief period, genuinely supportive of proper scientific process. The vice president of research convened a small internal review committee in March of 2009, consisting of Sandra, a physical anthropologist from the university's anthropology department named Dr. Marcus Fielding, and an external reviewer, a population geneticist from the University of Calgary, whose name I will not use here because he has since retired and asked not to be associated publicly with this account. This committee spent 4 months reviewing my raw sequence data, my extraction protocols, my chain of custody records, and my phylogenetic analysis methodology, essentially conducting an extremely thorough, extremely slow peer review process internal to the university before any external submission would even be considered. I want to be fair to that committee because they did their job properly. They found no methodological flaws. They independently reran my phylogenetic analysis using their own software and reference datasets and arrived at essentially the same divergence estimates I had calculated. Doctor, Fielding, the physical anthropologist, was initially the most skeptical member of the committee and became, by the end of that review period, the person most convinced that the genetic evidence was sound and that the appropriate next step was careful, controlled publication in a peer-reviewed journal, structured to establish the existence of the lineage without disclosing precise location data beyond the general watershed level, a compromise that Dr. Fielding himself proposed and that I believed, at the time, represented a reasonable middle path between full scientific disclosure and the protective discretion that Raymond had asked of me. The problems began in earnest once the review moved beyond that internal committee and into the layers of the university's relationship with outside government and funding bodies. The Ministry of Environment official who had been briefed early on, a regional director whose name I will withhold, took a considerably more aggressive interest in the project than any of us had anticipated, framing it internally, as I later learned through a colleague who saw the relevant correspondence, not primarily as a biodiversity discovery, but as a potential land use and resource development liability, given that formal documentation of a new, genetically distinct hominid population in a region with active forestry, mining, and pipeline development interests would almost certainly trigger environmental review processes that could delay or complicate existing and proposed industrial projects across a significant swath of north-central British Columbia. By the fall of 2009, what had begun as a scientific review process had become something closer to a negotiation between competing institutional interests, with my own research increasingly treated as a liability to be managed rather than a discovery to be properly investigated and eventually published. I was asked in a meeting, I remember, with particular clarity because it was the first time I understood how far this had moved from anything resembling normal scientific process, to provide precise GPS coordinates for every sample collection location, framed as a requirement for proper environmental assessment documentation by an official who, as far as I could tell, had no background in wildlife biology or conservation genetics, and whose primary concern appeared to be establishing a complete geographic data set that could be cross-referenced against existing and proposed resource development tenures in the region. I refuse. I want to be direct about that because it is the decision that, more than any other single choice I made during this entire period, defined everything that followed. I told the review committee and eventually the university administration directly that I would provide watershed-level location data sufficient for any legitimate scientific or conservation purpose, but that I would not provide precise GPS coordinates that could be used to locate specific individuals or family groups within the population, both because I had made commitments to Raymond and Colin regarding the protection of that information, and because I did not believe, as a matter of basic conservation ethics, that a newly documented population with an apparently very small total number of individuals should have its exact locations placed into any government or industry database regardless of the stated purpose, given the well-documented history of supposedly protected location data eventually finding its way into public records requests, leaked databases, or simple institutional carelessness. This refusal did not go over well. Sandra, who had been broadly supportive up to that point, began to distance herself from the project, telling me privately in a conversation in her office in November of 2009 that I have never fully forgiven her for, that my position was, in her words, not how institutional science works, and that I was putting both my own career and the university's relationship with its major funding and regulatory partners at risk over what she characterized as an excessive, and she implied, somewhat paranoid concern for information security that was not standard practice in wildlife genetics research. I want to pause here and say something in Sandra's defense because I have had years to reflect on this and I do not think she was wrong about the institutional realities, even though I believe I was right about the ethics. Sandra was not a villain in this story. She was a department head trying to protect her department's funding relationships and her own standing within a university administration that was, by 2010, actively pressuring her to bring the project into line with what the ministry and, increasingly, an outside pharmaceutical and biotechnology research consortium that had somehow become aware of the project's existence were requesting. I never learned exactly how that consortium learned about the work, though I have my suspicions and I do not think it matters much now. What matters is that by the spring of 2010, I was no longer simply managing a disagreement about data disclosure with my own university. I was managing pressure from multiple outside institutions, each with their own interest in either the genetic material itself, given the potential commercial and scientific value of a previously undocumented hominid genome, or in controlling the narrative around a discovery that had significant implications for land use and resource development across a large area of the province. I did eventually learn a little more about that consortium's interest, though not until years later through a conversation with a former colleague who had moved from the university into private biotech consulting and who mentioned, almost in passing over dinner in Vancouver in 2014, that a firm he had briefly contracted for in 2009 had been evaluating what he described only as a novel hominid genome of undisclosed origin for potential commercial applications in comparative genomics and pharmaceutical research, work he understood to be connected to a university project somewhere in the north of the province. He did not know it was mine, and I did not tell him, but the timeline matched too precisely to be coincidence, and it confirmed something I had suspected, but never been able to prove during the actual dispute: that the genetic material itself, entirely apart from any question of species conservation or scientific priority, had commercial value to interests well outside anything I had ever considered when I began this project as a straightforward question of unmatched mitochondrial sequences in 1996. That realization, arriving years after the fact, only reinforced my conviction that I had made the right choice in refusing to hand over location data, because a population whose genome had attracted commercial interest from a biotechnology consortium was a population that needed protection from collection pressure, not increased accessibility, regardless of what conservation language any government database might have used to justify requesting the coordinates in the first place. I have often wondered in the years since whether the ministry official who pushed hardest for that data in 2009 and 2010 understood the commercial dimension of what he was requesting, or whether he was simply a bureaucrat executing a data request that had originated somewhere else entirely, in an office I never saw and a set of interests I was never permitted to fully understand. I suspect I will never know the answer, and I have made my peace with that uncertainty in the same way I have made my peace with most of the unresolved questions this project left me with. The formal breaking point came in September of 2010 at a meeting I was called into with almost no advance notice, attended by Sandra, the university's vice president of research, a lawyer from the university's general counsel office, and by video conference, a representative from the Ministry of Environment, whom I had not met before and whose title I never fully understood, described to me only as being involved in interagency coordination. I was informed in language that was carefully neutral and carefully non-threatening on the surface, but unmistakable in its actual meaning, that the university had decided the project could not proceed toward publication under the current data disclosure terms, that continued research funding for the project would be contingent on my agreement to provide complete location data to a joint university government data repository with restricted but not zero access for name, research, and regulatory personnel, and that my continued employment, while not explicitly tied to this decision in the meeting itself, would be evaluated in the context of my broader research productivity and institutional cooperation during my upcoming tenure review, which was scheduled for the following academic year. I understood exactly what that meant. I had watched enough colleagues work their way through university politics over 15 years to recognize a threat delivered in the careful, deniable language of institutional bureaucracy when I heard one. I asked for time to consider the terms. I was given 2 weeks. I spent those 2 weeks doing something I had not done in 15 years of quiet, careful research. I stopped thinking about the science and started thinking seriously and specifically about what would actually happen to Raymond's trapline country, to the small population of beings whose existence I had spent a decade and a half proving if precise location data entered a government database that, regardless of stated access restrictions, would exist as a permanent record subject to freedom of information requests, staff turnover, data breaches, and the simple erosion of institutional discretion that happens to every sensitive government. data set eventually, given enough time and enough people with access to it. I thought about what would happen if a mining company's environmental consultant doing due diligence on a proposed exploration tenure gained access to that data through a formal interagency data sharing agreement, which was exactly the kind of arrangement the ministry official at that September meeting had described as standard practice for sensitive wildlife location data in areas with active resource development interest. I talked to Raymond during those 2 weeks, driving out to Smithers to see him in person rather than discussing it over the phone. I told him everything, including the pressure I was under and the choice I was facing. Raymond listened in the quiet, unhurried way he listened to everything, and then he told me something that settled the decision for me more than any amount of my own deliberation had managed to. He said that his father had told him when Raymond was a young man first learning to run the family trap line that there were places in that country that were not meant to be mapped, not because the land itself was sacred in some abstract sense, though it was that too, but because some things only survive by staying unmapped, and that the moment you draw a boundary around something and hand that boundary to an institution, you have already decided whether you meant to or not that the institution's claim to that place matters more than the thing living there. I went back to the university at the end of those 2 weeks and told the review committee that I would not provide precise location data under any circumstances, that I remained willing to publish a scientifically rigorous paper documenting the lineage's existence at the watershed level, sufficient to establish priority of discovery and to trigger appropriate conservation status review without enabling precise targeting of specific locations, and that if the university was not willing to support publication under those terms, I would need to consider my options outside the institution. The response was not immediate. It took another 4 months, through the winter of 2010 into early 2011, of what I can only describe as a slow institutional freeze. My research funding for the project was not formally canceled, because canceling it would have required an official rationale that the university did not want on record. It was simply deprioritized, delayed, and eventually left to lapse through a series of administrative decisions that were individually defensible and collectively devastating. My lab technicians were reassigned to other projects. My requests for continued access to the university's genomics sequencing facility for this specific research line were quietly denied on the grounds of facility capacity constraints that I had no way to independently verify or contest. And in March of 2011, during what should have been a routine tenure review process, I was informed that my case would require additional evaluation given what the review letter described as concerns regarding research transparency and data management practices identified during a recent internal review, language that referred, without ever saying so directly, to my refusal to hand over location data on a 15-year research project into a population that, as far as I knew, no one at the university outside the small original review committee had ever actually laid eyes on. I want to tell you about what happened next, because it is the part of this story that I think matters most, more than the DNA sequences, more than the institutional politics, more than the tenure review that eventually, in June of 2011, concluded with a decision that fell just short of outright termination, but that made my position at the university untenable in every practical sense. A formal reprimand for what the university characterized as inadequate documentation practices, a permanent note in my personnel file, and a public statement released without my input and over my formal objection describing the unidentified hominid research project as an unfunded, methodologically unverified side project that did not reflect the university's official research priorities or standards, language carefully crafted to distance the institution from 15 years of work while avoiding any direct claim that the underlying science was wrong, because they knew and I knew that the science had already survived one round of genuine, rigorous, internal peer review and had not been found wanting. I want to describe the tenure review meeting itself in more detail than I have so far, because it is the single conversation out of everything that happened over those 2 difficult years that I still replay most often, usually late at night, in the particular way that a person replays the exact sentence that ended something rather than the slow accumulation of pressure that led up to it. The meeting took place on a Tuesday afternoon in June of 2011 in a small conference room on the third floor of the administration building at the Prince George campus, a room I had sat in perhaps a dozen times before. Over 16 years for entirely routine departmental business, which made the formality of the seating arrangement that day, 3 university representatives on one side of the table and me alone on the other, feel considerably more adversarial than any of those previous meetings ever had. Sandra Whitlock was there, though she said very little for most of the meeting, sitting with her hands folded in front of her in a posture I had come to recognize over the previous 2 years as the posture she adopted when she had already decided an outcome and was simply waiting for the formal process to catch up to it. The vice president of research, a man named Dr. Alan Petrovich, whom I had worked under for most of my career at the university and who had, in earlier years, been genuinely supportive of ambitious, unconventional research directions in other departments, chaired the meeting. And a representative from Human Resources, whose role, as far as I could tell, was primarily to ensure that whatever was said in that room would not later expose the university to a wrongful dismissal claim, sat at the end of the table taking notes I never saw. Dr. Petrovich opened by summarizing the tenure committee's findings, reading from a document that used the phrase pattern of non-cooperation with institutional data governance protocols, 3 separate times in the space of about 4 minutes, language so carefully sanitized of any reference to what the actual underlying dispute concerned that a person walking into that room without prior knowledge of the project would have had no idea we were discussing a 15-year genetic research program into an undocumented hominid lineage rather than a routine administrative compliance failure. I asked him directly, partway through his summary, whether the committee's concerns were about the scientific rigor of my work or about my refusal to disclose precise location data to a joint government repository, and I watched him choose his words with visible care before answering that the committee's concerns were, in his phrasing, holistic, which I understood correctly as an admission that he did not want to answer the question directly on the record. I made my case as plainly as I knew how. I told them that in 16 years at the university I had published in respected journals, brought in consistent grant funding for legitimate wildlife genetics work, trained graduate students who had gone on to their own successful careers and never once had a finding of research misconduct or data falsification attached to my name, and that the entire basis of the committee's concern rested not on any failure of scientific method but on a disagreement about who should control the precise geographic coordinates of a small, vulnerable population that I had spent a decade and a half proving existed, largely through my own unfunded time and effort. I told them that I considered the protection of that location data to be not an obstruction of proper research governance, but the single most important ethical obligation attached to the entire discovery, and that I would not change that position regardless of what it meant for my employment. Sandra spoke only once during the entire meeting, near the end, after Dr. Petrovich had finished outlining the committee's proposed resolution, which fell short of termination but stripped the tenure decision of anything resembling an endorsement and effectively signaled that my future at the university would be, in practice, if not in official language, untenable. She said, looking at me directly for the first time since we had all sat down, that she wanted the record to reflect that she believed the science was sound, that she had reviewed it as carefully as anyone in that building had, and that her disagreement with me had never been about whether the discovery was real. It was, she said, about what an institution owes to the government and industry partners who fund it, and about the fact that I had never fully accepted that a university, whatever its stated mission, is also an institution with obligations that do not always align neatly with the obligations an individual researcher might feel toward the population he studies. I did not agree with her then, and I do not agree with her now. But I have thought about that distinction often enough in the years since to understand that she was not being dishonest with me in that room. She was describing accurately the actual structure of the institution we both worked within, one I had spent 15 years quietly hoping I could work around rather than confront directly. I left that meeting, drove the 4 hours back to Prince George from the meeting that had actually been held by video connection at a satellite administration office, and told Joan everything before I had even taken my coat off. She listened the way she always listened without interrupting, and when I finished, she asked me only one question. She asked whether I could live with myself if I gave them the coordinates. I told her no. She nodded, poured us both a drink, and said, "Then I suppose we're moving to Terrace." I resigned in August of 2011, 6 months before my 65th birthday, several years earlier than I had planned to retire. Joan and I sold our house in Prince George and moved to Terrace, closer to the coast, closer to Raymond and the country that had occupied the last 15 years of my professional life. And I spent that first year of what was technically retirement doing the thing I had never had the freedom to do while employed by an institution with its own competing interests. I went back through every sample, every field note, every phylogenetic analysis, and I prepared on my own time, under my own name, with no institutional affiliation attached, a manuscript for independent publication. I need to tell you what happened when I finally submitted that manuscript, because it is not the ending you might expect, and I think the honesty of that ending matters more than a tidy resolution would. I submitted the paper in the spring of 2012 to a respected mid-tier journal specializing in molecular phylogenetics, choosing a venue on purpose with rigorous peer review standards, but without the kind of high public profile that a journal like Nature or Science would have brought, because I had come by that point to want careful scientific scrutiny far more than I wanted public attention. The paper went through 3 rounds of peer review over the following 8 months. 2 of the 3 reviewers, both anonymous as is standard practice, raised substantive methodological questions primarily around sample size and the confidence intervals on my molecular clock divergence estimates, all of which I was able to address through additional analysis and clarification of my existing data, and neither of whom expressed the kind of reflexive dismissal I had half expected given the extraordinary nature of the claim. The 3rd reviewer recommended rejection on grounds that were, reading between the lines of a carefully worded review, less about the science itself and more about what the reviewer characterized as insufficient physical specimen evidence to support species description under formal taxonomic naming conventions. A fair methodological point, since I had, by design, no complete physical specimen, only hair, scat, and 1 set of plaster track casts alongside the genetic data. The paper was eventually published in a revised form that stopped short of formal species description, and instead presented the findings as documentation of a genetically distinct, undescribed hominid mitochondrial lineage in north-central British Columbia, with full methodology, full phylogenetic analysis, and a careful, tightly restrained discussion section that raised more questions than it answered, and that, crucially, specified location data only down to the watershed level, exactly as I had insisted throughout the entire ordeal with the university. The paper's publication in late 2012 did not produce the reaction I think most people would expect. There was no immediate media firestorm. There was no expedition of eager researchers descending on the Bulkley Valley. There was instead a fairly muted response within the small population genetics research community, some genuine scientific interest from a handful of colleagues who reached out privately to discuss methodology and potential collaboration on further sampling, and a great deal of dismissal from the broader scientific establishment, who treated the paper's subject matter as sufficient grounds to ignore its actual methodology, exactly the outcome I had spent 15 years trying to avoid by being as rigorous and conservative as I possibly could at every stage. A handful of popular science and cryptozoology outlets picked up the story, predictably sensationalized it in ways that made me deeply uncomfortable, and then, as these things do, the news cycle moved on within a matter of weeks. I want to describe 2 specific reactions from that period because they represent, I think, the 2 poles of how this work has been received ever since, and because both of them mattered to me a great deal in very different ways. The first came from Dr. Marcus Fielding, the physical anthropologist who had served on the university's internal review committee back in 2009 and who had, throughout the entire ordeal that followed, remained privately supportive even after he retired from the university himself in 2013. He wrote to me shortly after the paper's publication, a long letter on paper rather than by email, which he explained was intentional given how much of my recent history had been shaped by institutional data management that he no longer entirely trusted. He told me the paper was, in his professional judgment, the most carefully documented and methodologically conservative claim of its kind he had ever encountered, and that he considered it a genuine scientific tragedy that the institution that had trained me and employed me for 16 years had ultimately proven unwilling to stand behind it. He also told me, in a passage I have returned to many times since, that he believed history would eventually be kinder to this discovery than the immediate scientific community had been, though he was honest enough to add that he did not expect either of us would live to see that vindication arrive. The second reaction came from a science journalist at a national newspaper, whose name I will not include here because I do not believe she would want to be associated publicly with a story that her editors ultimately declined to run in the form she originally proposed. She interviewed me by phone for nearly 3 hours in January of 2013, having read the published paper carefully enough to ask genuinely informed methodological questions rather than simply chasing the sensational angle. And she told me near the end of that conversation that she found the science compelling, but that she doubted her editors would run a serious piece on it because the subject matter alone would trigger a set of institutional reflexes at the paper that no amount of methodological rigor could overcome. She was right. The piece never ran, or rather a much shorter, considerably more skeptical version ran eventually, buried in a weekend science section that treated the paper as a curiosity rather than as the substantial body of evidence it actually represented. I do not blame her for that outcome. I understood by that point exactly the institutional dynamic she was describing because I had spent the previous 4 years living inside a nearly identical version of it myself. Joan read every draft of the manuscript before I submitted it, and she read every piece of correspondence that came in afterward, both the generous and the dismissive, with the same careful, unsentimental attention she had brought to the sequence alignments spread across our kitchen table back in 2003. She never once suggested that I should have handled the university dispute differently, though she has told me more than once in the years since that she wishes I had found a way to bring her into full confidence about the project years earlier than I did, rather than protecting her, as I told myself I was doing at the time, from a burden I had convinced myself was mine, alone to carry. I think she was right about that, and it remains the one part of this entire 15-year project that I would genuinely do differently if I had the chance again. Not the science, not the refusal to disclose locations, not even the decision to resign, but the years I spent keeping the woman I had married in the dark about something that was quietly reshaping both our lives, one careful, secret evening at a time. I have had in the years exactly the outcome I think I should have expected, given everything I understood about institutional science by the time I resigned. The paper exists. It is citable. It is, as far as I know, still the only peer-reviewed genetic documentation of this lineage in the scientific literature. And I take some quiet satisfaction in that, even though it did not produce the broader recognition or the conservation protections I had once hoped a discovery of this magnitude might generate. I am not currently affiliated with any university. I do occasional consulting work for the provincial government on unrelated wildlife genetics projects, work that has nothing to do with the Telkwa lineage and that I suspect is offered to me in part as a quiet, unstated acknowledgement from people within the ministry who know exactly what happened to my career and who have never said so directly. I am not currently affiliated with any university. I do occasional consulting work for the provincial government on unrelated wildlife genetics projects, work that has nothing to do with the Telkwa lineage, and that I suspect is offered to me in part as a quiet, unstated acknowledgement from people within the ministry who know exactly what happened to my career and who have never said so directly. There is one more relationship from the university years that I want to close the loop on, because I think leaving it unresolved would give a misleading impression of how I have come to feel about the people involved in the institutional side of this story. Sandra Whitlock and I did not speak for nearly 6 years after I resigned, which I understood at the time as a mutual, unspoken agreement that there was nothing more productive either of us could say to the other. She retired from the university herself in 2017, and I heard about it secondhand through a colleague who still had contacts in the department rather than from her directly. She wrote to me in the spring of 2018 a short letter that arrived at the house in Terrace with no return address I recognized until I opened it and saw her name at the bottom. She told me she had spent a considerable amount of her retirement thinking about the way the tenure review process had unfolded, and that she wanted me to know, plainly and without qualification, that she believed the university had failed both the science and its own stated principles in how it had handled the situation. She wrote that she had come to regret her own role in that failure more than almost anything else from her career, not because she believed she had lied to me at any point. She still maintained, and I believe her, that everything she had told me about institutional obligations and funding relationships had been true as far as it went, but because she had allowed those institutional pressures to override what she now recognized with the clarity that sometimes only comes with distance and retirement as the more fundamental obligation a scientist owes to sound methodology and to the people like Raymond whose trust had made the underlying research possible in the first place. I wrote back. We exchanged perhaps a dozen letters over the following 2 years, careful, considered letters of the kind that people who have hurt each other and are trying to find their way back to something honest tend to write. And in the spring of 2019, a few months before Raymond passed, Sandra drove out to Terrace and Joan, and I had her to dinner, the first time the three of us had been in a room together since that conference room on the 3rd floor of the administration building 8 years earlier. It was not an easy evening. There were long silences, and there was a moment over dessert when Sandra apologized directly to Joan for the strain the entire ordeal had put on our marriage. An apology that surprised me more than almost anything else that evening, because I had never told her at the time or since quite how close that strain had come to costing me something I valued even more than the career itself. I do not want to overstate what that reconciliation accomplished. It did not restore my tenure or the 16 years of institutional standing I walked away from, or the funding that could have supported a far more thorough investigation than the one Raymond, Colin, and I managed to piece together largely on our own time and our own resources. But it gave me something I had not realized I still needed, which was confirmation from the one person inside the university who had been present for every stage of the institutional failure, that I had not imagined the scale of what had been asked of me, and that the choice I made had been recognized eventually by at least one of the people who had once stood on the other side of that conference room table as the only choice a scientist with any real conscience could have made. Before I get to how the story ends, I want to describe the years between the paper's publication in 2012 and Raymond's death in 2019, because they were, in their own quiet way, the best years of this entire project, even though almost nothing about them would make for a dramatic account. I was no longer racing against an institutional clock. I was no longer managing the tension between what I owed the science and what I owed the people who had trusted me with it, because that tension had already resolved itself one way or another by the time I left the university. What remained was simply the work itself, done for its own sake, and the friendships that had grown up around it. Joan and I settled into the house in Terrace in the fall of 2011. And I spent that first winter doing something I had genuinely missed during the busiest years of the project, reading widely outside my own narrow specialty, corresponding with colleagues in paleoanthropology and population genetics who had reached out after word of the university dispute had circulated in the careful, understated way that news travels through academic circles, through conference hallway conversations and shared acquaintances, rather than through any formal announcement. Most of those colleagues were cautious in their outreach, unwilling to attach their own names too closely to a project that carried the reputational risk mine did, but a handful were genuinely, quietly generous with their time, reviewing drafts of my manuscript, suggesting improvements to my molecular clock calibration methodology, and in 2 cases, offering to co-author follow-up papers using additional samples I continued to collect after leaving the university, work that appeared eventually in 2 smaller journal articles in 2015 and 2017, refining the population size estimates and extending the documented range slightly further north into the Skeena headwaters based on 2 additional samples Colin was able to collect during his final years with the ministry. Raymond's health began to decline in earnest around 2016, when he was already past 80, though he never fully gave up the trap line until the winter before he died, handing the registered license over to a nephew who had been shadowing him on the line for the better part of a decade, and who, Raymond told me with evident satisfaction during one of our last conversations, had already started noticing things in that country that he could not quite explain the same way Raymond himself had decades earlier, before either of us had any language for what those things actually were. I visited Raymond regularly during those final years, not for samples, which had stopped being the point of our friendship long before then, but simply to sit on his porch, drink coffee, and talk about the country we had both spent so much of our lives in the unhurried way that two old men who have shared something extraordinary and never needed to explain it to each other tend to talk. He passed away in the winter of 2019 at the age of 84, still living in the house outside Smithers where he had raised his family and run his trapline for most of his adult life. I was one of perhaps 30 people at his funeral, and I remember standing at the edge of that gathering, a retired university scientist who owed the most significant discovery of his career to a trapper's decision to trust him with a bag of hair instead of taking it to a newspaper, and understanding more clearly than I had at any point during the previous 2 decades that the actual center of this entire story had never been me. It had been Raymond and the generations of people before him who had known about that country and what lived in it long before any university existed to grant or withhold credentials for the privilege of confirming what they already knew. His family asked me to say a few words at the small gathering afterward, held at the community hall rather than at a church, and I have thought often in the years since about how difficult it was to find language for what Raymond had meant to this project without saying anything specific enough to violate everything he had spent his life protecting. I spoke instead about the kind of man he was in more general terms: patient, exact, generous with his knowledge but never careless with it, a man who had walked into my office in 1996 as a complete stranger and who had, over the following 23 years, become one of the 2 or 3 people whose judgment I trusted more than my own. His daughter, Elaine, who now holds the family's registered trapline alongside her son, thanked me afterward and told me that her father had spoken about our work together more often and with more evident pride than she had ever expected a man of his generation and temperament to speak about anything. That is, I think, the closest thing to a professional accolade I have received in the years since I left the university, and it means more to me than the publication itself ever has. I still go back into the Telkwa Range when my knees allow it, usually once or twice a summer now, sometimes with Colin Presley, who retired from the ministry a few years after I left the university, and who has become, over the years, less a professional contact and more simply an old friend who understands without needing explanation why a person might want to spend a few days each summer in country that most people will never know holds what it holds. I do not collect samples anymore, not in any formal sense. I do not need to. The genetic question, as far as 15 years of careful, methodical work could answer it, is settled, at least to my own satisfaction and to the satisfaction of the small number of colleagues who took the time to actually read the published paper rather than react to the subject matter. What I do on those trips now is simply walk that country the way Raymond taught me, to walk it quietly, patiently, without the need to prove anything or collect anything, and I have, in the years since I stopped trying to build a scientific case and started simply visiting a place I had come to love, experienced things I never recorded in any notebook and never will. Because some evidence I have come to believe was never meant to end up in a database at all. A shape at the tree line in the last light of a summer evening, gone by the time I raised my binoculars. A vocalization unmistakable after 25 years of learning to recognize it, carrying across a basin at dusk while I sat by a small fire, answered faintly from somewhere much farther up the slope. Once, 3 summers ago, a set of fresh tracks crossing directly over my own boot prints from earlier that same day, close enough to my camp that whatever made them had walked within perhaps 40 meters of where I slept and had chosen, as it apparently always had, simply to observe and pass by rather than to make its presence more directly known. I want to tell you about the most recent trip in July of last year because I think it says more about where I have landed with all of this than any summary of the science ever could. Colin and I hiked in together taking the same logging spur we had used for decades, though the spur road itself had grown over considerably in the years since active harvesting moved further west, forcing us to park nearly 2 kilometers further out than we used to, and adding an extra hour to what had once been a straightforward approach. My knees are not what they were even 5 years ago, and Colin, 3 years younger than me, has his own list of complaints about a body that spent 40 years doing field biology in mountainous terrain. So we took the climb slowly, stopping more often than either of us would have needed to 20 years earlier, and talking, as we always do on these trips, about everything except the thing we had actually come to think about, until the country itself made that impossible to avoid any longer. We made camp below the basin on the first evening, in almost exactly the spot where Raymond and I had camped on that first overnight trip in 2002. More than 2 decades earlier, a coincidence neither of us had planned, but that Colin noted quietly as we set up the tent, in the way that two old men noticed the accumulation of years without needing to say much more about it. The evening was clear, cold enough that we both had our jackets on by the time the lights started to go, and we sat by a small fire, not because we needed it for warmth, so much as because a fire is its own kind of company on a night like that, and we talked eventually about Raymond, about Trevor Adsit, whom neither of us has heard from in years, about the university which I have not set foot on in over a decade, and about whether either of us expected back when this all started that it would end up costing what it cost and giving what it gave in roughly equal measure. It was sometime after 10 the fire burned down to coals when we heard it. A single vocalization, distant, carrying down from somewhere high in the basin above us, low and sustained in exactly the register I have spent nearly 30 years learning to recognize without any conscious effort at this point, the way you recognize a familiar voice in a crowded room before you have even registered why. Colin went still. I did too. Neither of us reached for a recorder or a camera because neither of us has felt the need to document anything for our own sake in a long time now. And we simply sat there in the dark, listening until the sound faded and the basin went quiet again, except for the ordinary night noise of wind moving through subalpine fir and a creek running somewhere below us in the dark. Colin asked me, after a long silence, whether I ever regretted any of it, and I gave him the same answer I have given myself in one form or another, more times than I can count over the years since I left the university. I told him that I regretted the cost, the years of strain it put on my marriage before Joan and I found our footing again, the friendship with Trevor that never fully recovered, the 16 years of institutional standing I walked away from 6 months before I had planned to retire on my own terms. But I did not regret the choice itself, because the choice itself had never really been a choice in any meaningful sense. Not once I understood what handing over that data would have meant for the population I had spent a decade and a half proving was real. Some decisions only feel like decisions in retrospect. In the moment, they are simply the only thing a person with any conscience could have done, and everything that follows is just living with the consequences of having done it. I do not know how many individuals remain in that lineage today. My last population estimate, calculated from genetic diversity data before I left the university, suggested a minimum of somewhere between 15 and 30 individuals across the combined range I had documented, though the true number could be higher given how limited even 15 years of sampling was relative to the vastness of the terrain involved. It is not a large population by any measure. It is a population that has survived, as far as I can tell, precisely because it has remained hidden, protected less by any law or institution than by the sheer remoteness of the country it occupies and by the quiet discretion of the small number of people going back for generations before I ever arrived in that valley, who understood that some things are better documented than disclosed, better known than announced. I am 69 years old. I spent 15 years of my career proving something that the institution I worked for ultimately could not or would not protect the way I believed it deserved to be protected. And I lost a great deal in the process. Funding, standing, the tenured position I had spent a career building toward, and, for a difficult stretch in 2011, very nearly my marriage, though Joan and I found our way through that year together, the same way we found our way through everything else about this strange, difficult, extraordinary chapter of our lives. I do not regret the choice I made. I regret that the choice was necessary at all, that an institution built in principle to pursue knowledge for its own sake proved so much more interested when the knowledge in question became genuinely inconvenient in managing liability and controlling access than in protecting either the science or the population the science had revealed. What I have, instead of a completed career, instead of the recognition that 15 years of rigorous work might, in some better version of this story, have earned, is something I did not expect to value as much as I have come to value it. A friendship with a man who is gone now, but who trusted me. From the very first bag of hair he handed me across my office desk in 1996 to do right by something he had spent his whole life quietly protecting. A body of data that exists, published, peer-reviewed, permanent, regardless of how few people ever chose to take it seriously. And a handful of evenings each summer sitting by a small fire in a basin that very few people will ever walk into, listening to a sound in the dark that I spent 15 years proving was real and that I no longer need anyone else to believe in. I know how this account will be received by most people who hear it. I have made my peace with that a long time ago. I did the work, I kept the records, I published what I could publish without betraying the trust that made any of it possible in the first place, and I walked away from a career rather than hand a government database the exact location of a population that has survived hundreds of thousands of years by staying exactly where it is, unmapped, unclaimed, and, for now, still safe. Joan asked me when I told her I was finally ready to record this account, why now after so many years of choosing silence over disclosure even in this smaller, more personal sense. I told her the truth, which is that I am 69 years old, that Raymond is gone, that Colin and I will not be making these trips into the basin forever, and that I would rather the record of what I found and what it cost and what I still believe about how it should be protected exists somewhere in my own words before I am no longer able to offer it in any form at all. I am not providing coordinates in this account, and I never will, regardless of how many people ask. That has never been the point. The point was always simply this, that something real, something ancient, something far more deserving of quiet protection than of a government database entry lives in a stretch of mountains most people will never walk into, and that 15 years of my life were, on balance, well spent in making sure the world would eventually have to reckon with that fact on terms that did not require sacrificing the thing itself in the process of proving it. I think about that fire in the basin more than I think about the paper, more than I think about the conference room on the third floor of the administration building, more than I think about any of it. Somewhere up that slope in the dark, something answered a sound it had no reason to answer, the way it always has, the way it did for a trapper who never asked for anything in return except that the country be left alone. I spent 15 years proving that something the world does not believe in is real, and I would make every one of those same choices again, gladly, for the sound of that voice carrying down through the timber one more time.